Good morning, good day, and good evening to all. Welcome and thank you for attending the second webinar in part 2 of our webinar mini-series hosted by the Immunoassay Platform Solutions team at MilliporeSigma. My name is Andy Arena. I'll be the moderator for today's presentation entitled High Throughput Biomarker Profiling for Immuno-Oncology Research Using Milliplex Assays. It is my job to ensure that we make the most out of your time today. We have two speakers for you, Jan Nikhamin with MilliporeSigma and Jenny Koenitzer from Bristol Myers Squibb. Before we get started, here are a few things that everyone needs to know. First is the timing. We have allotted 60 minutes for today's webinar. This will be taken up largely by the presentation, but we have 15 minutes saved at the end for Q&A. And that brings me to the second point. To further enhance your webinar experience, we've set up some interactives throughout the presentation. First is the question submission. This will be ongoing throughout the presentation. Make sure to keep an eye on the questions being submitted, and you can upvote questions that you like. At the end of the presentation, we will work our way through some of these with priority on questions with the highest number of likes. And don't worry if you ask a question late and it doesn't get any likes or few likes. We have a team of 10 field scientists that are quarantined in their homes itching to answer every last one of these. I promise we have the time to get to all of them, and since you're registered with us today, we can follow up this presentation with a document providing the answers to questions asked. We will not include any names, only the Q&A, so please ask away. And then second is the live polling. These won't be hard-hitting questions, but keep an eye out for the classroom icon in the top right of the presentation. That indicates that a poll is coming on that slide. And here is a practice poll that we can take right now. Who is working from home today? I know I am. My family of 5 are here in the house. We're staying productive and positive, but looking forward to better days for sure. So please answer. We will leave the polls open for a minute or two, or until all the answers slow down and no longer come in. We have 3 more polls planned for today. So again, keep an eye out for the symbol in the top right corner of the slides. And finally, as I said before, my name is Andy. And just before I turn this over to the speakers, I want to point out that we did have a third webinar originally scheduled for this coming Thursday. The topic was homebrew development, converting Milliplex assays to our ultra-sensitive single molecule counting technology. Fortunately, we had to postpone this presentation, and we are working on confirming a new date, so please stay tuned. Now, without further ado, please welcome our first speaker. Jan Nikhamin is one of our immunoassay platform specialists. He's based in Brooklyn, New York, and he has spent the last 9 years at MilliporeSigma supporting our immunoassay technologies and products in New Jersey. Jan's presentation will focus on immune checkpoint proteins and their role in immuno-oncology research. Take it away, Jan.
Hello, everybody, and welcome to our webinar. Thank you for that introduction, Andy. And yep, very much like Andy, my family is at home as well, itching to get out and be out in the field. But for the time being, we're grateful that you have the time to spend with us today. And for our mini introduction, we'll be talking about the immuno-oncology landscape and the panel that we have that is being used by Jenny and her team. Slide. Thank you. Of course, we couldn't do without a little bit of marketing, and we wanted to remind everybody that our Milliplex portfolio is the largest on the market and is always growing. We have panels for just about every species out there. As a matter of fact, the one picture that's missing here is coming soon, and that'll be the ovine or the sheep panel. And within those species, of course, we have panels that are specific to different research areas should you need them. And slide, please. Okay. But for the focus of today, and for a big topic in the lives of researchers, of course, cancer is the big one. And we know that there are generally 10 hallmarks of cancer. We know that generally our immune system does a very good job of protecting us. And anytime a cell kind of gets out of control, we have a whole team of immune cells that will put them right back where they belong, and everything is basically nice and happy. However, tumor cells get clever and they try to escape those controls. And over time, we have learned that there are immune checkpoint proteins that are expressed on the surface of both presenting cells and immune cells that interact to either enhance the response or downplay that response. And slide, please. So a lot of research has been done over the last few years where we've learned which molecules interact with the immune system and how they actually interact. What are the responses? Are they inhibitory? Are they enhancing? Are they co-stimulatory? Now, generally, we know that all of these are membrane bound. And we know that there are cytokines that are soluble molecules that are flowing in the system that interact with all the cells to have these interactions happen. However, during the cancer environment or the tumor environment, whether it's in the body or in the microenvironment itself, a lot of these molecules or these checkpoint or inhibitor molecules can be cleaved. And if they are cleaved, we now have the opportunity to measure their presence and perhaps get a little bit more insight into the disease state. Slide, please. And with knowing that disease state, it improves our understanding and our knowledge, and perhaps improves our understanding on how to treat said disease. So, MilliporeSigma has come up with a panel that we call the Immuno-Oncology Checkpoint Protein Panel 1. It is a 17-plex panel that includes our popular or common immune check molecules that have become solubilized. Now, some of these are, of course, co-stimulatory, like CD80, CD6. Some of them are inhibitory, and like the currently popular PD1, PDL1, and PDL2. One thing that I would like to highlight about this particular panel is that we have gone to lengths to make sure that while there is homologous regions among all three molecules of PD1, PDL1, and PDL2, we made sure that there is no antibody crosstalk or cross-reactivity, and all the detection of these is absolutely individual and actually correct. Slide, please. So, here's a representation of the various standard curves that are included in this panel. You can see that they're biologically relevant, of course. The sensitivity and the min DC is represented. We can detect, I would suspect, almost all the states. It is possible that some of the healthy states may not be detectable, but that is something that we'll have to, I guess, figure out as research goes on, and Jenny will talk more to it. But what I wanted to highlight here specifically is we have made sure that our intra-assay and inter-assay precision is very good. That way, your continuous studies and your ongoing research is all relatable to the previous experiments. Slide, please. So, what does this allow us to do? This allows us to look at different disease models and paint kind of like a fingerprint with these molecule measurements to understand what happens. So, for example, we can take serum levels in the breast cancer samples and measure these immune checkpoint inhibitor molecules. And we can see that some are elevated versus normal, some are not, and CD80 is undetectable in this particular case. But it doesn't necessarily mean that all cancer will present exactly the same profile. So if we look at the sepsis panel, which is next slide, this profile is a little different. Now what happens here, we know that sepsis by itself provides an immune suppression. And seeing here there are elevated immune suppressing molecules that are solubilized, we can understand better how sepsis works. This so far so good, I hope. So next slide. Well, it also happens that within a given cancer disease, we know that there are different stages for various cancers, of course. So here we took an example of 3 different breast cancer lines and had them in conditioned media and measured the release of these immune checkpoint molecules in the media. And you could see that among the 3 different lines, they have a slightly different presentation of the checkpoints. Now, is this related to the particular stage that the breast cancer is in? Quite possibly. But this gives us a better picture and better understanding on what is going on within each stage of the disease progression. Slide, please. And likewise, we can look at other disease situations. So here is a sample where we're looking at colon cancer versus healthy control patients. And we could see that things like STAM, PD-1, TLRs are elevated versus the healthy controls where these molecules are very low to non-detectable within the serum. And that tells us that these, the cells within the colon cancer trying to escape their control by cleaving or solubilizing these particular molecules. Slide, please. Now, the picture of the immune checkpoint inhibitor of the panel itself is just a nice little snippet of the information that we get. And for that, of course, we present you with complementary panels that can be used in concert with the immune checkpoint inhibitor panel. For example, we have the high sensitivity or CD8 panel, we have the cytokine receptor panel, and we have the Th17 panel, which all have similar but more specialized ideas about looking into other cytokine and chemokine signal release that would work in concert with the information we collect from the immune checkpoint inhibitor. And so we can combine together the information that we gather from one panel with the inflammatory or with biomarkers and get a complete picture of a disease state. Instead of like in the good old days where we would look for the Western blot and have just one particular gene and that would be our aha moment, now we get to see the big picture with all of these pieces of information put together. Of course, the questions don't end there, and coming to a laboratory near you soon, hopefully once the pandemic is over, the plan is for the summer, we will have a sequel to the checkpoint inhibitor panel, which will be panel 2. This will be a 31-plex that contains some of your other favorites and contain molecules that maybe you were looking to find, like CD40 ligand, perhaps OX40, perhaps MAKE A and B, granzyme B, some of the popular ones. And this will make a bigger picture even more detailed. Now, I understand there's a new poll currently live. Feel free to answer, but in the meantime, slide please. I turn this back to Andy, and thank you very much for listening to me for the last few minutes.
Well, thank you, Jan. Okay, while you're answering the poll, move on. I'm very pleased to welcome our second speaker. Jenny Koenitzer is an immunobiologist at BMS in Princeton, New Jersey. While at BMS, she is focused on small molecule and biologics drug discovery in the immuno-oncology space. This work focused particularly in the area of T-cell checkpoint inhibitors and myeloid cell-mediated immune suppression. One of her current responsibilities is overseeing the Biomarker Profiling team, which provides high-throughput sample testing to the entire BMS research community. Jenny's talk will focus on how the Luminex platform is used to support very diverse early discovery research at BMS, the focus on immuno-oncology applications, and some case studies for the Milliplex checkpoint panel that Jan just described. Jenny, the floor is yours.
Thank you very much, Jan, and thank you very much, Andy, for the introduction and the invitation to talk about our research at BMS. So I'm going to spend the next 25 minutes or so with you to share how we use biomarker profiling to advance our research at BMS. I will introduce our biomarker platform and the role that biomarkers play specifically in the early stage IO discovery space. I also want to share some experiences that we had specifically with the Milliplex assays and show you some case studies of how we apply them. Finally, I want to leave you with an outlook of our future plans for the biomarker profiling space. My group, as Annie has said, is based in New Jersey, and we serve as a central biomarker profiling hub for the early stage discovery needs at BMS. We routinely work with and receive samples from all of our US research sites, which are in New Jersey, Massachusetts, California, and Washington, as well as from our international collaborators around the world. In order to facilitate high throughput analysis, we have established a semi-automated and miniaturized process that is based on the XMAP technology. We run all samples that we receive in a custom in-house developed 384-well process, which facilitates 75% cost savings and increases sample capacity at least fourfold, while also reducing the sample volume needed for profiling significantly. We also make heavy use of a number of automation platforms, liquid handlers, and workstations, for example, to support the consolidation of samples from submitted sample plates into master assay plates, or the steps of sample washing, for example. We then have a dedicated fleet of reader instruments that, when we are not quarantined in our homes, run pretty much 24/7 to process these samples. Finally, our entire process is built on a custom internal IT platform that allows our customers to submit their profiling requests online, help us with inventory and study data management, and supports the generation of customized reports for the different research communities that we work with. My team currently runs biomarker panels for 5 different species: human, mouse, rat, dog, and cyno, and we support projects from the early exploratory to the early clinical phases. The wheel on the right-hand side is intended to give you a flavor for the types of studies that we routinely run. One category is mechanism of action studies, where we profile samples generated from cell culture, for example, co-cultures of tumors and immune cells, or ex vivo stimulated preparations of primary immune cells in whole blood. Another big focus area are PK/PD or efficacy studies for preclinical animal models, such as syngeneic or genetically engineered tumor mouse models, but also rodent models used for fibrosis, cardiovascular, or autoimmune disease. A third and ever-increasing bucket of our work falls in the realm of toxicity studies, including exploratory tox studies for new investigational drug applications. Things we look for often with the appropriate analyte panels include cytokine storm and immune activation, as well as liver and kidney injury. And the last category that we work on falls in the realm of exploratory biomarker identification and patient stratification, where we profile biobank samples, including those from internal clinical trials, as well as various disease population samples that we typically source from our academic collaboration partners, including the corresponding normal healthy volunteer controls. In order to be able to support this wide range of study types, we need a very broad coverage of analyte panels for the different purposes in therapeutic areas. We currently have over 600 analytes in active use, and around 60% of those are in various forms of Milliplex Panels. We also have an ever-growing list of new analytes in development, which can either mean that we are working to miniaturize existing panels to add to our capabilities, or to develop completely new analytes, mostly through a very, very fruitful collaboration with the Millipore team. The different panels that we use are broadly classified into innate and adaptive immunity, heart failure, fibrosis, and toxicity. The immunology and soluble checkpoint panels are the ones I will mostly be focusing on the remainder of the talk, as our focus is on immuno-oncology. I had mentioned that a lot of our remit in biomarker profiling at BMS is on high throughput, and as such, we have optimized the workflows to be able to handle up to 2,000 samples each week with sample requirements between 5 and 20 microliters per individual panel. A very large fraction of the sample types that we typically deal with are serum, plasma, or supernatants and lysates from either cell cultures or tissues. We do, however, also occasionally work with more exotic matrices such as azides, blister fluid, synovial fluid, bronchoalveolar lavage, or increasing the urine. We have been building this platform over the last few years, and its use across the BMS research community has really taken off. To date, we have profiled more than 235,000 samples for 170 different discovery programs, which resulted in more than 10 million data points. This large-scale profiling brings its own challenges in data management, which I will touch upon a little bit later. Now I want to switch gears a little bit and focus specifically on our immuno-oncology support. Of the roughly 47,000 samples that we handled last year, almost half of them, shown in red, came from immuno-oncology. As you can see in the breakdown on the right, the majority of the samples are from human origin, followed by mouse and cyno. On average, each individual study that we run has about 70 samples, but the range can be very wide, from as few as 10 samples to as many as 800 samples in a particular study. Similarly, we profile a mean of 41 cytokines for particular samples. Again, the range is very wide, from as little as 14, which would be an individual panel, all the way up to 100, which would be a number of different panels. Consequently, the average number of panels we run per sample is about 3. We have certainly seen over the last year or two that the demand for broader profiling, more analytes per sample, is ever increasing, which is in line with more and more analytes becoming available for different pathways. Now, you might ask yourself why we expend such a lot of effort to investigate soluble biomarkers in general and cytokines in particular for our IO research. It might become clearer when looking at cancer as essentially the result of a dysregulated immune system, as in Ira Mellman's and Dan Chan's cancer immunity cycle shown here, where every step of successful tumor eradication is dependent on cytokines as the master regulators, from tumor antigen presentation to the activation and priming of different relevant immune cells, to trafficking those cells to the site of malignancy, and finally infiltration, recognition, and destruction of the tumor cells. We have long known that cytokines play dual roles in tumor regenesis. They contribute to tumor formation, for example, by promoting pathogenic inflammation and tissue damage or invasion, metastasis and angiogenesis, but also play key roles in preventing tumor formation or resolving early-stage malignancies. Understanding this intricate balance of the immune system's master regulators is key in developing successful IO drugs, and the first step is understanding which molecules are present at what stage of cancer development. Indeed, cytokines themselves are also interesting therapeutic options for cancers. This graphic from a 2019 review shows which one of the cytokines have or are currently being investigated in clinical trials, from interferons to GM-CSF and various interleukins. Indeed, interferon alpha and IL-2 are already approved treatments for hairy cell leukemia and melanoma and renal cancer, respectively. And both of those treatment mechanisms basically rely on broad immune stimulation. With this outside role of soluble biomarkers, including cytokines, chemokines, and checkpoints, it is probably not surprising that we have a fairly large selection of analytes in routine use for our IO-focused projects. Here I am showing the 133 analytes and their corresponding panels that we use for human profiling of IO samples at BMS. We have similar, somewhat smaller selections that we use for mouse and cyno studies. All of these panels that I'm showing here are based off existing Milliplex panels. And I want to use the next couple slides to show you some of our experiences with those panels. So when we do profiling for IO samples, we don't just use the checkpoint panel. Typically, it's usually a combination of various different panels depending on what type of question of innate or adaptive immunity we actually want to answer, how much volume we have for a particular sample, and what sort of sensitivity do we need to get the data. So my first example for our experience with the Milliplex panels goes back to the sensitivity and quantification in the high sensitivity Milliplex panels. So sensitivity, in particular, the lower limit of quantification, are critical aspects for a lot of the cytokine biomarkers, as a large fraction of those biomarkers are only found in very, very low concentrations, in particularly when using unstimulated samples or healthy controls. We have frequently struggled with detecting certain biomarkers reliably in different sample types, let alone properly quantify them. That is why we were very eager to adopt the high sensitivity panels when they became available, and are always on the lookout for new high sensitivity analytes being added to this selection. The tables that you can show on the slide, on the left for human, on the right for mouse, show the comparison of the LLOQ, the lower limit of quantification, compared between the Milliplex normal regular sensitivity panels that we initially started using and the newer high sensitivity panels. The LLOQs in our case are determined by the fit of the respective analyte standard curve under our specific assay conditions. As you can see, most of the analytes have a vastly improved LLOQ, in some cases very significantly by 50 or more fold. Some analytes for which this has been especially helpful are IL-10, IL-12, P70, IL-4, 5, and 6, where we routinely use these high sensitivity analytes to get a better understanding of our samples. Now, the LLOQ is a particular function of how we run our assays and how we run our standard curves. So your individual mileage, depending on how you run your assays, may vary. But in our hands, they have performed very, very well. My next example for working with the Milliplex panels comes to adopting the checkpoint panel 1 that Jan had introduced. So when the checkpoint Milliplex panels or checkpoint Luminex panels in general became available on the market, we set out to figure out which ones behaved well under our particular assay conditions. And these are, of course, of key use when we profile patient samples for patient stratification. So this 70 analyte soluble checkpoint panel from Milliplex is now in pretty wide use, as it includes the key markers like PD-1, PD-L1, PD-L2, and LAG-3. And we are certainly very excited to later this year test checkpoint panel 2 as well. When we originally wanted to adopt checkpoint panels, we wanted to see how they performed. As we have done a large variety of work on targeting checkpoint molecules in-house at BMS, we have a number of recombinant proteins available to test a particular kit's performance. Exactly this type of data is shown on this slide in heat map format, and what I'm showing is the raw fluorescence intensities from this particular experiment. Across the x-axis, you can see the different analytes or signal channels and bead regions. Along the y-axis are the respective standards that come with the kit at the top, followed by the recombinant proteins PD-1, PD-L1, and PD-L2 that we spiked into the samples. You see that as expected, across the top, each standard produces a nice dose response dependent signal in it with its respective bead. Going down the list, then you'll notice that in our hands, the specificity of the kit is excellent. Each recombinant protein only produces a signal in the channel, or with the bead where we would expect it with. Everything else looks like background, giving us confidence in this particular reagent. At the time, we were also evaluating other kits that are available in the market. And here I'm showing an example of one that doesn't behave quite as well. Same concept as before, a slightly different analyte composition in the kit. The kit standard, by and large, behave well, and the control proteins produce strong signals with their respective beads in our condition. However, under our assay conditions with this kit, we also observed significant cross-reactivity with several beads, which are kind of the blue arrows where I tried to highlight where we got a signal where we wouldn't expect it, which of course then means that we ultimately can't reliably quantify the samples with regards to the individual checkpoint amount. Now, for the next section of my talk, I want to focus on some key applications of biomarker profiling in our IO discovery research pipeline. And I see there is a poll online, so please go ahead and answer if you would like. And I also want to share on the following slides some case studies to give you a flavor of how we use cytokine profiling data and multiplex data to progress individual projects. The case studies basically encompass 3 different flavors of experiments that we support. First are in vitro cell models, such as profiling the signatures of primary immune cells or tumor cells, as well as co-cultures. We use these experiments typically very early in a project during the target validation phase. The second type of experiments are in the preclinical or in vivo group, where we profile tumor models in animals, either in vivo or ex vivo, and also analyze samples from exploratory tox studies, often performed in cynomolgus monkeys for a particular drug candidate. And the third and final category encompasses all types of patient samples. These are typically the samples that are the most precious and where we perform the most possible broad analysis. In the early stages of an IO discovery project, we use a number of complex cellular models to analyze a particular target's or compound's behavior. One of these models that we use widely is the co-culture of genetically engineered primary T cells, which express a known antigen-specific T cell receptor with a matched tumor cell presenting the cognate antigen on their MHC molecules. That way we can co-culture these 2 cells and measure antispin specific activation and ultimately tumor cell killing by the T cells of the tumor cells. An example of that is shown in the pictures on the right, where the tumor cells are labeled with a red dye and we measure cell death with a green dye staining apoptotic cells that then externalize annexin 5. And the top picture basically shows the absence of antigen in the co-culture, which means the T cell cannot recognize the tumor cell. There is no activation and the tumor cells basically overgrow the well. In the bottom case, we have added the antigen to the co-culture, and now the tumor cells can be recognized by the antigen-specific T cell, be activated and killed, which means during the co-culture, you can actively monitor the lysis and the killing and basically the disappearance of the red tumor cells and the increase of clustering of dead cells in those green clusters that I'm showing there, the right-hand side on the bottom. Also, T cell activation results in a very strong increase in T cell specific cytokine production. So we frequently use the Milliplex T cell panel to quantify those changes in a higher throughput. And that is an example that I am showing here, where the whole concept of this particular project is to turn a cold tumor that has no antigen presentation into a hot tumor that presents antigen and then enables antigen-specific T cell activation and cytokine production. And as I have mentioned, we use the T cell panel shown on the bottom right very widely to run these types of experiments. And we model this particular project by co-culturing different types of tumor cells with the antigen-specific T cells, with or without molecules that act as epigenetic modulators, which we hope will induce antigen expression. And this is data from an actual experiment shown in the heat map in the middle. And the way I have represented the data is as a log two-fold change of the individual cytokines measured over the unstimulated control. And in this case, unstimulated control means a tumor T cell co-culture where no antigen is present, so no T cell activation occurs. Across the bottom, you see 3 different flavors of tumor cells that we have used. Let me start on the left-hand side where we used a wild type tumor cell. And we basically have a positive control. We load that tumor cell with the cognate antigen, which leads to strong T cell activation and ultimately tumor lysis, which is indicated by the green column of numbers on the first-hand side of on the most left-hand side of the table. So this basically means that the T cells were very, very strongly activated and lysed the tumor cells and proliferated and produced all of those different molecules, including, for example, granzyme or interferon gamma, which are key for mediating T cell effector function. Then, instead of actually providing the antigen to the co-culture, we instead treated the tumor cells with epigenetic modulator 1 or 2, to see if interfering with several of these pathways, we could achieve the same effect, so induce antigen expression in a tumor that normally does not express this antigen. And as you can see, for the epigenetic modulator number 1, we actually do get a good response. It seems like the tumor cell is now, after treatment, able to be recognized by the T cell, and from other data, we know that it does indeed now express the antigen and present it on its MHC molecule. Epigenetic modulator number 2, on the other hand, didn't really work that well. We wanted to make sure that this observed behavior was indeed due to the tumor cell antigen presentation pathway, which needs a whole machinery from gene expression to protein production and processing of those proteins to the eventual loading of the antigenic fragment onto the cellular MHC models before a T cell receptor can recognize the tumor cell. Therefore, for this project, we created some CRISPR knockouts of key proteins in those pathways. And those are the other 2 cell lines you see across the bottom in purple and in blue. We made a beta-2 MKO. The beta-2 MKO is an essential component of the MHC complex on the cell surface. So in the absence of this protein, no functional MHC complex can be formed, and the cell is not capable of presenting any antigens, whether they come from outside or inside of the cell. The second is a TAP1 KO. So TAP1 is an essential transporter for intracellular antigen processing and antigen loading onto the MHC. So in the absence of this protein, those particular cells are not able to present newly expressed neoantigens, but are expected to have a functional MHC on the surface. And indeed, our cytokine data using the antigen-loaded control confirm what we expected. The tumor cells without a functional MHC at all, the beta-2 MKO, no longer induces T cell activation, even when we load the antigen externally. However, the TAP1 KO still has an MHC and is therefore able to perform like the wild type. In both cases, our epigenetic modulators don't work anymore due to the deficiencies in the antigen presentation pathway, which together with other data gave us confidence that the approach is promising. So this is an example where we use complex cellular models and routine profiling of T cell activation markers to better understand the mechanisms and validate targets in IO. My next case studies come more from the mouse in vivo work that we do, specifically from an orthotopic transplant model of pancreatic cancer. Here, the specific question was if radiation therapy, a standard of care approach, can enhance immunotherapy efficacy of PD-1 inhibitors or combinations of PD-1 and IDO inhibitors. Ultimately, the data revealed that survival was improved by a combination of radiation and anti-PD-1, but that IDO combinations were not useful, but that the efficacy of the PD-1 activity correlated with a systemic interferon gamma response that we monitored by multiplex cytokine profiling. As my third example, I want to give you a flavor for how we use cytokine profiling to evaluate the toxicity risks of our later stage molecules before filing an IND. In this particular example, a BMS molecule was given in increasing doses. Group 1 is the control group, and group 2 through 4 received the drug twice a week apart, and we received serum samples for cytokine profiling at different time points, from just before the first dose until about 2 weeks after the last dose. Here we assessed a total of 44 different cytokines in 2 different cyno-specific panels. I have just chosen 5 relevant ones for the line graph, and each line in the graph represents an individual monkey. We observed a dose-dependent transient increase in inflammatory cytokines that peaks 3 to 6 hours after dosing and returns to baseline within 24 hours. We also saw that the second dose doesn't cause the same magnitude of effect. Together with other data that was collected from the study, we could conclude that the molecule was well tolerated and did not cause a cytokine storm, which was a particular concern for this particular mechanism of action of this molecule, because we could also, from in vitro data we had previously generated, figure out that these transient cytokine inductions that we see are most likely on mechanism and on target. In my last example, I want to talk to you very briefly about our work with patient samples. And this is where we most broadly apply the checkpoint panel 1 that Jan had introduced. When we obtain these types of patient samples, we typically perform very, very broad profiling, as much typically as sample volumes permit, often for 100 or more analytes, including the checkpoint panel. So the heat map exemplifies an example of what such data could look like. This aggregates several hundred serum samples by different tumor types, which shows clear differences in the checkpoint molecules detected. Ultimately, the goal is to build a sufficiently large database that allows us to identify predictive biomarker signatures when linked to patient phenotypic or treatment data. And another focus for us is also performing longitudinal studies on treatment, particularly with anti-PD-1, to investigate the response to treatment or the development of resistance mechanism and their impact on the respective biomarker signatures. Further, what we also frequently do is use explants to evaluate individual treatments and treatment combos. And this biomarker data, the multiplex Luminex data, is typically one key readout of those types of studies. With that, I hope I was able to give you a flavor for how we use multiplex biomarker profiling in IO discovery at BMS, and I want to finish with an outlook on our current and future focus areas. An ever ongoing effort is the expansion of coverage, so adding new panels of analytes to the platform or developing new analytes altogether in close collaboration with Millipore. We also have a growing need for custom disease-specific panels to support targeted biomarker studies. And we also are always very interested in trying to consolidate and merge individual panels wherever possible to both reduce workloads and the volumes of sample that are needed. Finally, we also spend a lot of effort in 2 closely related areas, data analytics and data integration. I have shown you that we profile tens of thousands of samples a year across a wide variety of experimental designs. With that comes a strong need for data management and, more importantly, metadata management, so all the data can be applied for the widest possible use. So we are constantly trying to optimize our data registration and database models or build new systems to handle these types of data. This is also key in facilitating enhanced bioinformatics methods for data analysis, as well as efforts to implement machine learning, particularly for generating disease predictive biomarker signatures. Finally, biomarker data becomes much more informative when it can be overlaid with other data types generated from the same types of studies, such as sequencing or phenotyping data, for example. So we are trying to build structures that enable to do that more easily. And lastly, an effort that we have ongoing focuses on new technologies that allows investigating the cytokine profile of a single cell. As we know, immune cells in particular show very heterogeneous responses to treatment. And ultimately, we want to marry that single cell data to the bulk biomarker data that we routinely produce from the Luminex profiling. With that, I want to thank you for your attention and acknowledge, and particularly the BMS cytokine team, especially Melissa Yard and Nathan Cheadle, who are the engines of this platform and keep the sample and data flowing on a day-by-day basis. And we are also indebted to all of our BMS collaborators, both those who enable us to physically run all the instruments that we need, as well as the partners in the individual therapeutic areas who supply us with the samples. And I also want to acknowledge our very strong partners at Millipore, particularly Jan and Shane, who we meet with frequently in non-pandemic times, as well as Brooke, who supports our ever-expanding list of requests for new analytes. And with that, thank you very much again, and I'll hand it back over to Andy.
All right, thank you, Jenny. Thank you, Jenny and Jan, for this great presentation.